Hey everyone, welcome back for today's deep dive. I'm excited to jump into some really fascinating genomics news in the last 48 hours.
Yeah, there have been some big developments.
Absolutely. So consider this your cheat sheet to stay ahead of the curve in the world of genomics.
Definitely some need-to-know breakthroughs.
Let's start with aging research, shall we? There was a fascinating study published in Nature Aging from researchers at UC San Diego.
Oh, yeah, this one really caught my eye. It looks like they may have found a crucial link between two of the most prominent theories on aging.
Exactly. For years, scientists have been debating whether aging is primarily driven by random genetic mutations that accumulate over time,
Or by these more predictable epigenetic changes that alter gene expression as we age.
and this study seems to bridge those two ideas in a way that's never been done before.
It really does. They found specific mutations in a gene responsible for DNA repair that directly affect epigenetic changes.
So essentially, damage to our DNA repair system could actually speed up the aging process.
Precisely. It's like a double whammy for aging.
Wow. So what are the implications of this for potential anti-aging therapies?
Well, this discovery opens up some really exciting possibilities. If we can target these mutations or the downstream effects they have on epigenetics.
We could potentially slow down the aging process itself.
Exactly. Imagine therapies that could either repair those DNA repair genes or mitigate those epigenetic changes they trigger.
That's incredible. It's like we're on the verge of being able to directly target aging.
It's definitely a game changer and something we'll be following closely.
For sure. Now let's shift gears to another major development, a significant funding boost for diversifying human genome research.
This is huge. Washington University in St. Louis just received 2 grants from the NIH totaling $14 million.
$14 million. That's a serious investment. So what will this funding be used for?
It's all going towards the Human Pangenome Reference Sequencing Project.
Right, and just as a reminder for our listeners, why is this project so important?
Well, the current reference genomes that scientists use are largely based on European ancestry.
Which doesn't accurately reflect the diversity of the human population.
Exactly, and that limits our understanding of how genetic variations impact health and disease across different populations.
It's like trying to understand the whole world by only looking at one small piece of it.
A perfect analogy. This project aims to create a much more comprehensive and inclusive reference genome that represents the full spectrum of human genetic diversity.
So what are the potential benefits of having this more inclusive pangenome?
Oh, the possibilities are vast.
Oh.
More accurate diagnoses, better prediction of disease risk, and even tailored drug development for a much wider range of people.
It's a crucial step towards making sure everyone benefits from the power of genomic medicine.
Absolutely. This is about equity in healthcare and ensuring that everyone has access to the best possible treatments based on their unique genetic makeup.
Absolutely. Now for our final story today, let's take a quick look at the RNA-seq blog and see what's buzzing in the world of RNA sequencing.
The RNA-seq blog has been highlighting some really interesting research lately, especially in the field of single-cell analysis.
I saw that there were a few articles about new sequencing techniques that caught my eye, but some of those titles were a bit of a mouthful.
Haha, yeah, they can get pretty technical. But essentially, these advancements are allowing us to understand what's happening inside individual cells with incredible precision.
So let's try to break down a few of those for our listeners. One that stood out to me was SmartSeq plus 5 Radar sheds light on transcriptional regulation during early mammalian development.
Ah, yes, that one's fascinating. This technique allows scientists to study how genes are activated in the very earliest stages of development.
So we're talking about understanding the very first moments of life at a molecular level.
Exactly, which could lead to breakthroughs in understanding and maybe even preventing birth defects and developmental disorders.
Wow, that's incredible. Another post I saw was about sequencing technologies to measure translation in single cells.
Right. Now, this is all about seeing how cells build proteins in real time.
So it's like watching the cellular machinery in action.
Precisely. This kind of insight could be invaluable for understanding diseases caused by errors in protein synthesis.
Like certain cancers or neurodegenerative disorders?
Exactly. And then there was that article about UDA-seq, universal droplet microfluidics-based combinatorial indexing for massive-scale multimodal single-cell sequencing.
Yeah, that one was a real tongue twister. Can you unpack that one for us?
Sure. Basically, it means that we can now analyze thousands of single cells simultaneously. Imagine the possibilities for studying complex tissues and uncovering rare cell populations.
That's incredible. It opens up a whole new world for research and targeted therapies.
It really does.
Uh,
We're moving from looking at cells in bulk to understanding the unique roles of individual cells in health and disease.
It's amazing to see how rapidly the field of RNA sequencing is progressing.
It is. And with each new discovery, we're getting closer to unlocking the secrets of human biology at its most fundamental level.
That's an exciting time to be following genomics, for sure.
Absolutely. The future of medicine is being written right before our eyes.
And it really underscores the importance of that push for diversity in genomics research we were talking about.
Oh, absolutely. The Human Pangenome Project is going to be crucial for making those kinds of individualized treatments a reality.
Because without a truly diverse reference genome, we're essentially working with an incomplete picture of human genetics.
Exactly. And that can lead to missed opportunities for diagnosis, treatment, and even drug development for certain populations.
Right. It's about ensuring that everyone benefits from the power of genomics, not just a select few.
It's like if you were trying to build a house but you only had half the blueprints, you might end up with something that's structurally unsound or doesn't meet the needs of the people who live there.
That's a great analogy. And these advancements in RNA sequencing we talked about, they really open up a whole new level of precision in medicine.
Absolutely. We're moving beyond just treating a disease in general to targeting the specific cells that are driving that disease.
It's like having a molecular scalpel to go in and surgically remove the problem at its source.
Precisely. And with these new techniques, we can actually see how individual cells are responding to treatment in real time.
It's like having a personalized dashboard for each patient, showing you exactly what's happening at a cellular level.
Exactly. And as these technologies continue to improve, we can expect even more precise and effective treatments in the future.
It's really an exciting time for genomics and medicine. But as we delve deeper into this cellular world, it also raises some important ethical questions.
Oh, for sure. With great power comes great responsibility, as they say.
Absolutely. We need to make sure these advancements are used ethically and equitably for the benefit of all humanity.
One of the biggest challenges is going to be ensuring access to these technologies for everyone, regardless of their background or socioeconomic status.
Right. We can't allow genomics to become a luxury that's only available to the privileged few.
Exactly. We need to make sure that everyone has the opportunity to benefit from these advancements.
Another crucial consideration is data privacy and security. As we generate more and more genomic data, we need to make sure that information is protected from misuse.
Absolutely. This is especially important as we move towards personalized medicine, where individual genomic data will be used to make treatment decisions.
We need to find the right balance between sharing data for scientific progress and protecting individual privacy.
Exactly. It's a complex issue, but one that we need to address head-on.
And we also need to think about the broader societal implications of these advancements.
Right. How will our understanding of genetics change our perceptions of ourselves and others?
Exactly. And how will it shape our decisions about things like reproduction, healthcare, and even our identities?
These are big questions that we need to start grappling with now.
As we stand on the cusp of this incredible new era in genomics.
It's an exciting time, but also a time for careful reflection and thoughtful dialogue.
Yeah, it really feels like we're entering this uncharted territory in genomics and medicine.
It is, and as we uncover more and more about our genes and how they influence everything from our health to our behavior,
We need to be really mindful of the potential consequences.
absolutely. One of the most important things to consider is access to these technologies.
Right. We can't let genomics become a luxury that's only available to the wealthy or privileged.
Exactly. We need to ensure that everyone benefits from these advancements, regardless of their background or socioeconomic status.
It's about creating a more equitable future for healthcare,
Right.
where everyone has the opportunity to reach their full potential.
Absolutely. And that also means being very careful about how we handle all this genomic data.
Right. Data privacy and security are paramount, especially as we move towards more personalized medicine.
Exactly. We need to strike that delicate balance between sharing data to advance scientific progress and protecting individual privacy.
Because ultimately, it's about empowering individuals with knowledge about their own genetics without compromising their rights.
Exactly. And as we learn more about the complexities of the human genome,
We also need to be prepared for the potential societal implications.
great. How will this knowledge impact our understanding of ourselves and others?
Exactly. And how will it shape our decisions about things like reproduction, healthcare, and even our identities?
These are profound questions that we need to start addressing now.
Because the answers will have far-reaching consequences for generations to come.
It's a fascinating and challenging time to be alive, that's for sure.
It really is. We're on the brink of a genomic revolution that has the potential to transform healthcare and our understanding of what it means to be human.
And it's up to all of us to ensure that this revolution benefits all of humanity.
Well said. That's a perfect note to end on. Thanks for joining us for this deep dive into the world of genomics.
It's been a pleasure as always.
Remember to check out the show notes for links to all the research we discussed today, and be sure to tune in next time for another fascinating glimpse into the future of genomics.
Until then, keep exploring and stay curious.